today's topic is translation. As we have already done transcription in the previous lecture, today we are going to deal with translation. So what is translation? According to central dogma, the process of conversion of RNA into protein is known as translation. There are various units involved in this process. For example, mRNA, tRNA, ribosomes, and amino acid, tRNA synthetase enzyme, as well as ATP. We will study all these units one by one. For example, first is mRNA or messenger mRNA. When DNA changes into RNA, it contains various exposing nucleotide sequences. When we combine these nucleotide sequences in a pair of 3, these are known as codon. Why it is necessary to study codon? Because these are the units which will help in the attachment of different amino acid or to form a new or unique polypeptide chain. This varies from organism to organism. Second unit we are having is tRNA. It is also known as soluble RNA. First of all, let us study how it is formed, then we will study its structure. This is RNA. When it undergoes coiling, it forms a tertiary structure such that again and again coiling of this RNA form a particular shape that is known as tertiary structure. We can represent this structure in a simpler form like this, that is, it is a T-shape or clover leaf shaped structure. What is clover leaf? Leaf which have 4 parts. Similarly, it has 4 parts. It actually has 5 arms. That is one which is variable loop or arm, that is 3D in nature. It is not represented in the front direction. In the 3D direction, it is just behind the structure. So its arms are, this is D loop that help in the attachment with the enzyme, T loop that help in the attachment with ATP. This is amino acid acceptor arm where amino acid will be attached, and this is anticodon loop which will code opposite to the mRNA. We can also study this in the form of L-shaped structure. This is amino acid arm and this is anticodon loop. This is mRNA, and according to which complementarity it is will be containing the nitrogenous bases. Next is ribosome. As we have already studied in our previous classes about ribosome, that it is a protein factory in a cell and it has 2 parts, larger subunit and smaller subunit. Actually, its structure in this process look like this. It has 3 sites in the larger subunit, that is A site, P site and E site. From the A site, this tRNA get attached in. At the P side, there is formation of polypeptide, and from the E side, the tRNA moves out of the process. Units involved are amino acid, tRNA synthetase enzyme, and ATP. Now let's study the steps involved. The first step is initiation. That is, in that too, A step is activation of amino acid. To transfer amino acid to the tRNA, we need to activate it first. How we will activate it? By using the energy currency, that is ATP or adenosine triphosphate. When any amino acid gets the ATP, it gets activated in the presence of enzyme and it will form a AMP enzyme complex, that is amino acid monophosphate enzyme complex. Here we are presenting the amino acid by methionine, ATP by a black dot, and the enzyme by this symbol. So here we'll get this kind of complex. In the next step or the B step, that is transfer of amino acid to tRNA. Here this complex will be added with the tRNA. Now this enzyme will transfer this activated amino acid to tRNA. Therefore, this tRNA will receive that amino acid on the amino acid acceptor end, and we will get adenosine monophosphate and enzyme. They will separate out, and this is now our charged tRNA. C step is assembly of units involved. The 5 units we have studied till now, they will assemble together to start the process. Here in this process, the mRNA will be first attached by the smaller ribosome unit that will attach to a particular point from the 5' to 3' strand, and this tRNA, charged tRNA, will attach to the AUG codon in the mRNA. Next, there is the attachment of larger subunit. The larger subunit attach in a way that the amino acid comes at the P site. As I have already told you, there are 3 sites. The charged tRNA, first that is AUG or the initiator codon, its charged tRNA will have UAC codes at the anticodon loop and methionine at its amino acid acceptor arm. Step is elongation. In this step, another charged tRNA that is complementary to the mRNA sequence present will come and get attached at the A site, and the P site tRNA will move to the E site in this way, and while moving out of the E site, the tRNA will give its amino acid to the previous or the coming tRNA at the P side, thus forming a chain, or we can say a dipeptide is formed. Similarly, it will be released from the E side and another tRNA will come from the A side according to the sequence present in the mRNA. This process continues for several times until we obtain a required amount of polypeptide chain. Step is termination. In this, the last codon or the stop codon UGA, to which attaches our release factor, that does not code for any amino acid, thus it stops the whole process and terminating the process, and the units disintegrates in this format, that is larger subunit, smaller subunit of ribosome, mRNA, last tRNA, release factor and a polypeptide that we are forming, or the protein.